This module pertains to mitochondrial DNA analysis. After learning this module, you shall be able to understand structure and inheritance of mitochondrial DNA, mitochondrial DNA and its forensic importance, analysis of mitochondrial DNA and interpretation of results. Commonly used short tandem repeats or STR typing system cannot be used every time. Often degraded DNA samples does not produce proper results, but from environmentally damaged DNA samples, DNA information can be recovered with mitochondrial DNA. Although mitochondrial DNA cannot give better results than nuclear DNA, but it is still used as it is better than no results at all. If the DNA is extracted in low amount, mainly from tissues such as bone, teeth and hair, then the chances of getting results are higher in case of mitochondrial DNA as compared to nuclear DNA. Mitochondrial DNA is present in higher copy number in cells as compared to nuclear DNA. Hence, they can be recovered from degraded sample as well. Mitochondrial DNA is present in hundreds of copies, whereas only 2 copies maternal or paternal of nuclear DNA are present in each cell. As regards its forensic issues, since mitochondrial DNA is present in higher amount and can be recovered from degraded DNA as well, it plays an important role in forensic testing, which involving sample with limited biological material. Mitochondrial DNA can be used in missing persons investigation and even in mass disasters. Since mitochondrial DNA is inherited only from mother, the siblings or any maternally related individual will have similar mitochondrial DNA sequences. Therefore, by using reference sample from maternal relatives, forensic comparison can be made. These comparisons can be done even if reference sample and unknown samples are several generations apart. As regard characteristics and structure of mitochondrial DNA, mitochondria is present in the cytoplasm and is the energy producing cell organelle. It consists of a small and circular genome. The amount of mitochondrial DNA present in cell varies tremendously. Mitochondrial DNA has about 16,569 base pairs and 37 genes in human that code for products which are used in cellular energy production or oxidative phosphorylation. It also contains 1122 base pair region which does not code for any product and is known as non-coding regions. It is present in hundreds of copies in each cell. Amount of mitochondrial DNA is about 0.25% of the cell content and is packed in a circular chromosome which is haploid. It is not unique as compared to nuclear DNA and is identical in all maternal relatives. It has a high mutational rate. Mostly there are small mutations like insertions or deletions. Genes in mitochondrial DNA are packed with no introns and only a few non-coding nucleotides between coding regions. replication of mitochondrial DNA starts with H strand in non-coding region, and this non-coding region is also known as displacement loop or D loop. This is the figure showing the structure of mitochondrial DNA in humans. Inheritance of mitochondrial DNA, it is inherited strictly from one's mother. During fertilization, only sperm's nucleus enters the egg and fuses with egg nucleus. Other cellular components are not contributed by the fertilizing sperm. Therefore, mitochondrial DNA of female gets transferred to offspring without any male influence. In case any paternal mitochondrial DNA passes through the egg, it gets completely diluted due to presence of about 1 lakh molecule of mitochondrial DNA in the egg. Presence of ubiquitin tag during spermatogenesis destroys any of the sperm mitochondria which enters the egg. Since mother transfers her mitochondrial DNA type to her offsprings, all these siblings and mother have identical mitochondrial DNA. Hence, mitochondrial DNA is not unique to the individual. Since it is not unique, mitochondrial DNA can not be used for individuals identification during mass disaster or missing persons identification, but it can be used to reduce the significance of match in forensic cases. It is useful in solving historical puzzles and to link families. Analysis of mitochondrial DNA. There have been a progress in methods used for measuring mitochondrial DNA in the past decades. The methods are used to trace maternal genealogy. Low resolution restriction fragment length polymorphism analysis was used for the first time with mitochondrial DNA in 1980s, in which 5 or 6 restriction enzymes were used. PCR amplification of 9 overlapping fragments followed by digestion with 12 to 14 restriction enzyme was done for high resolution restriction analysis. Use of portions of control region of mitochondrial DNA came into wide acceptance in 90s for forensic DNA typing. Maximum mitochondrial DNA variation between individuals in human are found in D loop. Within D loop, there are two regions known as hypervariable region 1 and hypervariable region 2. These regions are first PCR amplified and then sequence analysis is done. About 342 base pairs of hypervariable region 1 and 268 base pair of hypervariable region 2, say total of 610 base pair are examined, and any difference in the nucleotide position is noted. Sometimes a region of 137 base pair is also examined. This region is also known as hypervariable region 3. In order to avoid contamination issues, reference sample should be analyzed after complete processing of evidence sample. Blood stains or buccal swabs are usually taken as reference sample which contain enough amount of high quality DNA. As regards sample extraction, mitochondrial DNA is mainly used when there is a less amount of DNA present. Commonly used materials for mitochondrial DNA analysis are teeth, hair and bone etc. In forensic cases, mitochondrial DNA is extracted carefully to avoid destruction of the physical feature of the bone. To extract the sample, only a small section of the bone is cut from the middle to keep the overall length of the bone intact. The same precaution should also be taken care in case of teeth, so that the teeth can be further used for odontological examination. Estimation of mitochondrial DNA is done like nuclear DNA quantitation assays. PCR amplification. For amplifying mitochondrial DNA, 34 to 38 cycle of PCR are used for its amplification. Sometime for highly degraded sample, about 42 cycles are also used. To overcome PCR inhibitors such as melanin, additional units of Taq DNA polymerase are added. DNA sequencing started through Sanger method developed over 40 years ago, which is still in use. A separation step capable of single nucleotide resolution is done after polymerase introduction of DDNTPs, dideoxyribonucleotide triphosphates, as chain terminator. Chain growth is terminated when dideoxy NTPs is attached with 3' end of DNA nucleotide and no hydroxyl group is attached at 3' end. Reaction mix contain extendable DNTPs and DDNTP terminators, which lead to extension of some portion of DNA. A series of molecules are present at the end of sequencing reaction, which have a difference of one base from another. Sequencing of each strand is done separately with a single primer. For this purpose, reverse or forward primer are often used. 4 different DDNTPs are labeled with 4 different colored fluorescent dyes. Red dye is used to label DD-TTP or thymine. Green dye is used to label DD-ATP, that is adenine. Blue dye is used to label DD-CTP, that is cytosine, and yellow dye is used for guanine. Fluorescent dye labels have simplified DNA sequencing. Brighter dyes have improved signal to noise ratios, which helps in obtaining results from less amount of material. For a DNA sequence reaction, about 1 nanogram of mitochondrial DNA PCR product can be used. Following steps are performed for mitochondrial DNA sequencing. Number 1, use of various primer sets for PCR amplification of entire control portion. Then spin filtration using enzymatic digestion with shrimp alkaline phosphatase and exonuclease 1 and a microcone 100 filter to remove remaining DNTPs and primers from PCR. PCR product quantity is determined. Incorporation of fluorescent DDNTPs is done and each reaction has different primer to see which strand is sequenced. Use of spin column filtration is used to remove unincorporated fluorescent dye from completed DNA sequencing. Use of capillary or gel electrophoresis to separate the formamide diluted purified sequencing reaction products. And next is interpretation of the compiled sequence information after sequence analysis of each and every reaction performed. The major difference between mitochondrial DNA sequencing and STR analysis on the basis of multicolor fluorescence detecting instrument is its separation medium, which is capable of single base resolution. It is not required for STR typing, but it is needed for mitochondrial DNA sequencing. As regards interpreting and reporting mitochondrial DNA results, first data review and editing is done. To maintain the quality, comparison of complementary strand is done by performing DNA sequencing in both the direction, that is forward and reverse. If sequence from both strand is obtained, then sequencing of the same strand is done twice in separate reactions. The main purpose of this is to have at least double coverage of every nucleotide. In recent year, there have been an improvement in sequencing instruments and chemistries which lead to better sensitivity, less noise, even more peaks. In spite of the advanced technology, the analyst should review each nucleotide manually using computer software, followed by editing of it if any base calling algorithm has made an incorrect call. Generation of alignment from multiple sequences which are generated over a region for a particular sample helps in editing process of sequence. Alignment of forward and reverse sequencing reaction is done using computer programs like sequencer, which allows sequencing of electropherogram for every reaction and helps in side by side evaluation. As a final quality assurance measure, examination, interpretation and editing of the matching sequences should be done by two forensic analysts independently. After completion of mitochondrial DNA sequence analysis, comparison of questioned and known sample is done using results of edited and reviewed sequences. Comparison of two sequences will give the result as perfect match and not a match. If sample match at each evaluated site, then they are termed as concordant. The results are generally grouped into three categories. Number 1 is exclusion. If there are two or more differences in nucleotide between questioned and known sample, then it can be interpreted that the samples are not from the same source. The second category is inconclusive. The results is considered inconclusive if there is a difference of one nucleotide between known and questioned sample. And the third category is failure to exclude or cannot exclude. If there is a match between bases at each position or common length variant in hypervariable region 2, see stretch of questioned and known sample, then it can be interpreted that the sample are from same source or maternal lineage. This table shows the sequence in questioned and known samples, the observation and then interpretation. In case of ambiguity, for example, heteroplasmy, that is presence of more than one mitochondrial type in an individual, a common base is known as shared base in the sequence. In this case, they cannot be excluded from each other. Since mutation have been observed in between mother and the child, the difference in single base is termed as inconclusive result. In case of inconclusive result, additional reference sample are obtained and sequenced to clarify the interpretation. In summary, it can be said that commonly used short tandem repeats typing system cannot be used in case of degraded DNA sample, but from environmentally damaged DNA samples, DNA information can be recovered with mitochondrial DNA sequencing. Mitochondrial DNA can be used in missing persons investigation and even in mass disasters. Since mitochondrial DNA is inherited from mother, sibling, an individual's mother or any maternally related family members will have similar mitochondrial sequences. Mitochondrial DNA is present in hundreds of copies in each cell and they are all inherited from mother only. Low resolution restriction fragment length polymorphism analysis was used for the first time with mitochondrial DNA in 1980s, in which 5 or 6 restriction enzymes were used. PCR amplification of 9 overlapping fragments followed by digestion with 12 to 14 restriction enzyme was done for high resolution restriction analysis. Commonly used material for mitochondrial DNA analysis are teeth, hair and bones etc. In forensic cases, extraction and purification of mitochondrial DNA from these samples is carried out very carefully. 34 to 38 cycle of PCR amplification are required for mitochondrial DNA sequencing. Sometime for highly degraded samples about 42 cycles are used. To overcome PCR inhibitor such as melanin, excess tag is added. DNA sequencing is carried out through Sanger method, a separation step capable of single nucleotide resolution is done after polymerase introduction of DDNTPs, that is dideoxyribonucleotide triphosphates as chain terminator. 4 different DDNTPs are labeled with 4 different colored fluorescent dyes. Red dye is used to label thymine, green dye is used to label adenine, blue dye is used to label cytosine and yellow dye is used for guanine. To maintain the quality, comparison of complementary strand is done by performing DNA sequencing in both the direction, that is forward and reverse. In case sequence from both strands is not obtained, then sequencing of the same strand should be done twice in separate reaction. The main purpose of this is to have at least double coverage of every nucleotide. If sample match at each evaluated site, then they are termed as concordant. Results are generally grouped into 3 categories, that is exclusion, inconclusive and failure to exclude or cannot exclude. In case of any ambiguity, that is for example heteroplasmy, presence of more than one mitochondrial DNA type in individual, a common base is known as shared base in sequence. In this case, they cannot be excluded from each other. Thank you.